Good morning or good afternoon, depending on where you're joining us from. I'm so glad to see all of you here today. My name is Dan Schimberg, and I joined the SRF Board of Directors earlier this year. In 2020, a close family member of mine was diagnosed with scleroderma. Since then, our family has been deeply committed to supporting the most promising research in hope of finding a cure for this insidious disease. We believe SRF is our best hope for that answer. I'm excited to introduce our next session, which explores a fascinating and forward-looking topic, cell therapy and scleroderma, exploring tomorrow's possibilities. This session is divided into two parts, but feel free to submit your questions for either speaker at any point along the way. First up, Dr. Ryan Crisman, co-founder and chief technical officer at Umoja Biopharma, and he will be presenting the next generation in cell therapy. Dr. Ryan Crisman is the co-founder and chief technical officer at Umoja Biopharma. Before starting Umoja in 2019, he helped to build and lead several labs focused on developing new cell and protein therapies for patients. His work has played a key role in bringing many first-in-human treatments to clinical trials, including efforts at companies like Juno Therapeutics, Zymogenetics, and the Gates manufacturing facility. Then we will hear from Dr. Elaine Karis, senior clinical trial physician at Bristol Myers Squibb, presenting CAR-T in systemic sclerosis, understanding data, inclusion criteria, and eligibility. Elaine completed her rheumatology fellowship at NYU Medical Center and practiced for several years before transitioning to clinical development with a focus on bringing innovative therapies to patients living with autoimmune diseases, including systemic sclerosis and systemic lupus erythematosus.
Welcome. Hi, I'm Luke Evnin, and I'm chair of the Scleroderma Research Foundation. I'm also your moderator for this session. I thought I'd add just a few preamble remarks to set the stage for two fantastic speakers who will be following me in just a minute. So, we are absolutely in the midst of a revolution in the treatment of autoimmune disease more broadly. Frankly, it was not so long ago that the only available therapy for the signature autoimmune disease, rheumatoid arthritis, was gold salts. This paradigm finally began to change in the late 1990s with the first approval of a biologic to block the TNF molecule. TNF blockers are now absolutely standard of care in RA, and frankly, they've been used all across autoimmune disease with very significant success. Unfortunately, not so much in scleroderma. In scleroderma, if we were to look back, say 20 years, the standard of care was deep immunosuppression with a drug called cyclophosphamide. Now, this therapy, with its fairly significant systemic toxicity burden, was ultimately replaced by MMF, mycophenolate. However, advances over and above mycophenolate have frankly been pretty hard to come by in scleroderma, even as better options have emerged for other manifestations of autoimmune disease. Now, recently, the idea that autoimmunity could effectively be eliminated and the diseased immune system of an autoimmune patient could be reset has gained currency. The promise of such therapy, which could be achieved through depletion of the entire B-cell arm of the immune system, has obvious allure. Now, just to set the stage again, about 10 years ago, work was done in scleroderma to show that chemoablation of a patient's immune system, followed by the rescue of that patient with the patient's own stem cells, actually did show significant disease improvements. However, the intensity of that therapy and the extensive hospital stay required by that, led to relatively modest adoption of that therapy. But here we are today to talk about some of the amazing progress being made in B-cell depletion therapy and to talk about the applications of that specifically in scleroderma. Just a couple more introductory points about B cells. Now, as you may know, they're the only cell that can produce antibodies, and those soluble proteins are absolutely essential in fighting off disease. Now, however, B cells also have another element. They're integral elements of the complex web in the immune system that signal back and forth. So there's direct interactions between another piece of the adaptive immune system, T cells, and the B cells. And also, B cells secrete soluble growth factors called cytokines, which have profound effects on other elements of the immune system. Okay, one more fact about B cells. B cells are made all the time. They're made in large quantities and they start from a precursor cell. Now, as B cells mature from their precursor cell to their ultimate mature antibody-producing self, they express different proteins on their cell surface. You'll hear a little bit about those later today. Some of the important ones are a cell surface molecule called CD19, another called CD20, and another called BCMA. These are all different proteins expressed on the cell surface of the B cell at different times in the maturity as B cells progress from precursor to mature antibody-producing cells. And with that, I'm delighted to introduce our first speaker, Dr. Elaine Karis from BMS.
Hello, everyone. I'm so glad to be here today to talk to you about investigating CAR T cell therapy in systemic sclerosis, seeking a fresh start for the immune system. So the immune system is the body's natural defense system. It defends against invaders, including harmful viruses, bacteria. It can even kill cancer cells. So some of the main defenders are the T cells and the B cells. These cells, they detect and destroy threats while sparing healthy cells. In autoimmune diseases, the immune system attacks healthy cells. And overactive B cells are culprits in autoimmune disease. These are the same cells that produce autoantibodies. So instead of attacking invaders, they attack healthy tissue and healthy cells that can cause damage to the body, which can get worse over time. So the goal of CAR T is to attack these overactive B cells. So CAR T cells are a type of cellular therapy. There are other cellular therapies that are being investigated currently, but for the purposes of this talk, I would like to focus on the CAR T cells. The CAR T cells are made from the body's own T cells. So the T in the CAR T is the person's own T cell. And the CAR is the chimeric antigen receptor that is added to the cell surface during a manufacturing process, so that the person's own T cell is then directed specifically to go kill B cells in their own body. Now, CAR T is not new, although it is new to the autoimmune space. It was approved in 2017 in the oncology space for the treatment of lymphoma. So in oncology, it is approved therapy. In autoimmune space, it is still an investigational treatment. So it's only available in clinical studies right now. So what happens in order to manufacture the CAR T is that the person's own blood is collected, which of course includes those T cells. That blood is then sent to a manufacturing facility where the CAR is added to the cell surface, and that CAR can vary from company to company, but is a message basically provides a signal to the cells about which B cells to go and kill. So once that CAR T cell is manufactured and available, the body needs to be prepared to receive the CAR T cell treatment. So for that preparation, prior to CAR T, people will receive chemotherapy. And that chemotherapy is just a pre-treatment, pre-infusion to prepare their body to receive the CAR T so that the CAR T can then function to its utmost potential. The CAR T cell infusion itself is just received as one infusion. And then, of course, people are monitored for potential side effects following the CAR T infusion, both for short-term and long-term potential side effects. So this slide shows the effects of the CAR T on the immune system. So once the lymphodepletion and the CAR T are received by the person, we see a very rapid decline in the B cells. The B cells are destroyed from the body. During this process, and shortly following it, there may be a risk of developing two adverse events specific for CAR T. Those are known as cytokine release syndrome and ICANS. People are very closely monitored for signs and symptoms of the development of these conditions, which are, well characterized based on our understanding in the oncology space. So we know what they are and we know how to treat them. But it is important that they are assessed and identified as soon as they occur. And they, of course, they don't occur in everyone. We don't yet know the likelihood of these events occurring in the autoimmune population because our current data is just so limited to date. But I can tell you some information from this study here. Then when another important point is that when those B cells are just depleted to that great extent, the CAR T is expected to result in a phenomenon known as immune reset. So immune reset is when you deplete all the bad B cells, as you can see here, followed by reconstitution of B cells. So the B cells do come back. So the thing about CAR T is, although patients are very heavily immunosuppressed when given the treatment, eventually, because this is a one-time treatment, it's not like we typically do in the autoimmune space where we chronically immunosuppress for months or years. In this case, you immunosuppress very heavily, and then those B cells are allowed to come back. So your immune system is actually allowed to come back. And when it comes back, what we're noticing is that from those mature pathogenic B cells, which are autoantibody producing and causing problems, what we see come back are a naive, fresh set of B cells, which can fight invaders, but are not programmed to fight your own immune system. So some additional evidence of that is here on the right-hand side, where you can see the autoantibodies, in this case, the anti-nuclear antibodies, which are very commonly seen in systemic sclerosis, and are also positive when people have presence of scleroderma-specific antibodies. We see that, and those autoantibodies are produced by the B cells. So when the B cells are depleted, we also see the autoantibodies are depleted as well. So this is what is happening behind the scenes in the immune system. Here, what we see are that in these 6 individuals treated for CAR T, we have some clinical improvement that was also reported. What we saw were that no patients in the trial worsened their systemic sclerosis. So that was the primary outcome of the study, was to see whether anybody worsened after CAR T, and no one worsened. And that was after a median follow-up time of 487 days, which is over 1 year. But some additional details were shared with us. So the modified Rodnan skin score, which is a test, pinch test of the skin over various areas of the body, which measures skin thickness, where a score of 0 is just a normal skin thickness, and 3 is very severe affected skin from systemic sclerosis. What we see here is that the modified Rodnan skin score, or that skin thickness, improved, again, very rapidly over the course of the first 6 months or so, and then continued to improve over the course of at least 1 year. And then on the right-hand side, they reported digital ulcerations. So for those patients that had digital ulcerations at baseline, they did see that the number of digital ulcerations did decrease by 3 and 6 months. And by 12 months, it appears that they were all gone. Next slide. So additionally, Raynaud's phenomenon, a very common manifestation of systemic sclerosis, was reported that the number of Raynaud's attacks per day did not change. But what did change was the duration of the attacks. So in these patients, they were having shorter Raynaud's attacks, and also the pain intensity that was associated with those attacks was also reported to improve. They also reported improvement in hand function. So this is also very important. This has to do with the dexterity of the hands, and the hands were shown to improve, and this might be a reflection of the improvement in the skin. It could be a reflection of an improvement in arthritis, but in general, we see the hand function is improved. And importantly, so I didn't talk yet about the baseline characteristics of these patients, but these patients were, systemic sclerosis patients. They were both male and female. They varied in age range. They all had the presence of autoantibodies, and they all had the presence of interstitial lung disease. So when they looked at the interstitial lung disease in these patients, what they noticed is that based on high resolution CT, so a CAT scan, which looks at the extent of interstitial lung disease and lung fibrosis, was able to show that there was actually a decrease in the extent of interstitial lung disease on the CAT scan. Additionally, patients had pulmonary function tests over the course of this study. And the pulmonary function test measured a forced vital capacity, which is a very common measurement of lung function. And forced vital capacity is typically compromised in patients with interstitial lung disease. And what they showed here was actually an improvement in the patient's forced vital capacity over time. So this is very encouraging. That there be a potential to improve lung function in patients with interstitial lung disease, as well as we saw on the prior slide, which is the potential to improve skin, as well as hand function and Raynaud's symptoms. They additionally reported and showed an image of a patient who had inflammation of the heart due to their systemic sclerosis. And when they did a follow-up image at 3 months, what they showed was also decreased inflammation in the heart. So for clinical trials of cellular therapy, again, there are many clinical trials underway. They all have some variety in their clinical trial design. Additionally, there is variety in the types of cellular therapy that is being used. So whether it be a T cell or an NK cell or a different type of a cell, as well as the CAR on the cell surface may differ. Very commonly, it is a CD19-directed CAR, which means that the patient's T cells, the CAR T, will go and kill B cells with CD19 on the surface. There's another type of CAR T known as a BCMA CAR T, and that one will go and kill B cells with BCMA on the surface. So we don't yet know, what the clinical differences may be with these different approaches. We're still better understanding the safety in these different approaches and the adverse events that may occur. But in general, there is depletion of the B cells and immune suppression with the lymphodepletion. So there will be a period of time where there is a risk of infection, although the B cells do get reconstituted, as we previously saw. So those B cells do have the opportunity to come back, and the immune system does have the opportunity to come back and be able to fight those other invaders, those bacteria and viruses, after some time. So I just wanted to talk here a bit about the differences in the populations that are included in the various clinical trials. In general, there are 2 different populations that are included. Some clinical trials are choosing to include patients with diffuse skin disease, so patients with extensive skin involvement, proximal to the elbows and knees, whether or not they have lung involvement. So these are basically studies in diffuse systemic sclerosis with or without interstitial lung disease. I would note that all the trials basically are excluding patients with limited skin disease if they don't have lung involvement. So in this group here with diffuse skin disease with or without lung involvement, the limited skin patients do not qualify whether or not they have lung involvement. This is studying diffuse systemic sclerosis. There are other trials that are studying patients with limited or diffuse skin disease. So the extent of skin involvement doesn't matter in those trials, but what does matter is the presence of interstitial lung disease. So in those trials, patients must have interstitial lung disease. If they don't have interstitial lung disease, they are not qualifying for those studies. As mentioned, in general, limited patients without interstitial lung disease are excluded from all CAR T trials that I am aware of that are occurring to date. Additionally, patients with really severely compromised lung function, so when that forced vital capacity or that diffusion capacity measured on the pulmonary function testing are very low, they are excluded from the current trials, and also if they are requiring oxygen therapy. Additionally, patients are excluded if they have severe or unstable cardiac disease. So if they've had a recent heart attack, if they have heart failure with symptoms, those patients are also typically excluded and likely to be excluded until we know more about CAR T and systemic sclerosis. And thank you so much for having me today and allowing me to give you some information about CAR T and systemic sclerosis. And please be on the lookout for additional information. As time goes on, we will see more and more data reported on CAR T in this disease.
Thank you, Dan, for that introduction, and thank you to Luke Evnin and the Scleroderma Research Foundation for the opportunity to be here and share my thoughts on the future of cell and gene therapies in scleroderma. I'm Ryan Crisman, and I co-founded Umoja Biopharma with the focus on how to solve patient accessibility and patient experience in the cell and gene therapy space. It's an extremely exciting time in that we're standing at the forefront of a paradigm shift in the way that we understand and treat autoimmune diseases. Traditionally, these diseases have been managed with broad immunosuppressants and symptom-focused therapies. And these complex conditions pose significant challenges both for the patients and the clinicians alike. But the landscape is changing, and it's changing quickly. Advances in cell and gene therapies are opening up entirely new avenues for precision treatment, targeting more of the root cause of the immune dysfunction rather than just managing the consequences. So things, from engineered T regulatory cells to gene editing approaches that recalibrate immune tolerance, we're really witnessing an emergence of therapies that promise durable and, I might even argue, potentially curative outcomes for diseases that were once considered intractable. And so these advancements really are being led in a significant part by the groundbreaking work in CAR T-cell therapeutics. And so what I've done here is tried to kind of bucket what I thought were some next-gen efforts being put forth both by biotech and big pharma. And as I did that, it started to come out that, of these 6 boxes that I highlight, 4 of them are in the CAR T space. And so the question is being, asked, well, why this sudden excitement in CAR T cell therapies in autoimmune disease? So I think if we look back just a short time ago, a couple of years ago, this really started initially out of the Schett lab and the early clinical data that he showed. And so what I'm highlighting here in a couple of call-out boxes is from his paper in the New England Journal of Medicine in 2024. Really, it was a CD19 CAR T-cell therapies in autoimmune disease. And really, this early data sparked what I would consider a renaissance in the CAR T therapy for autoimmune diseases. These results in autologous CD19-directed CAR T-cell therapy showed deep B-cell aplasia that was durable beyond 100-plus days with a single infusion of CAR T-cells. So huge promise with this technology, and I have to emphasize it's early data, but as we saw on the previous slide, companies are now flowing into the area. And really, if you think about it, there are a few key insights that we can take away from the early data. The first that I think about is safety. And if you read through the papers and read through the data, the procedure is shown to have acceptable safety in these small cohorts that we've seen. The safety is probably as good, maybe even better than what we've seen in the autologous CAR T-cell treatments for oncology. And from an efficacy perspective, the patients in these trials, they've demonstrated marked improvements in skin fibrosis and stabilization, or maybe even improvement in their lung functions and reduction in the systemic inflammation and the autoantibodies. And so from an efficacy perspective, again, seeing a lot of promise with the CD19 treatments in these small case studies. And then lastly, from a mechanistic perspective, this approach is thought to work by depleting the autoreactive B cells, essentially resetting the immune tolerance without the need for full myeloablation. So what I wanted to do then was how do these compare to the myeloablative autologous stem cell transplantations? And if we look at that landmark study that looked at cyclophosphamide versus transplantation, that data showed that this myeloablative autologous stem cell transplantation offers long-term benefits in severe systemic sclerosis compared to cyclophosphamide. The survival and the disease control in this study showed that stem cell transplants improved the event-free and the overall survival. It improved lung function. It improved quality of life. However, it came at a trade-off of toxicity, in that the high-dose chemotherapy needed came with significant acute toxicity and even had low single-digit treatment-related mortality. And so from a safety perspective, there was a pretty significant trade-off. I wanted to look at maybe the CAR T cells from a qualitative perspective, just because it's such early data in small numbers. But I tried to highlight a little bit about what I see as kind of the trends in towards the CAR T cells. If we look at from an immune response, again, for CAR T cells, these are looking at targeted B cell depletion as opposed to a broad chemoablation. Toxicity, we know that the stem cell transplants have high toxicity because of the high chemotherapy. CAR T cells, we anticipate, if we extrapolate based on the data that we have from the oncology, we anticipate it to be lower, but it's not zero. There's still low-dose chemotherapies for lymphodepletion to pre-treat the patients. We know that the CAR T cells themselves have some toxicity issues. We know how to manage them. So I would anticipate the toxicity to potentially be lower, but still not negligible. From a durability perspective, we know that there's great durability with the stem cell transplantation. And again, we're too early in the CAR T space to know if we're going to see similar durability and efficacy. But if we think about extrapolating again from the oncology data, it would suggest that we could see really good durability with patients on CAR T treatment. And then the last one, and something that I always think about, is kind of the patient experience, the patient burden. We know that high-dose chemotherapy is really, really challenging. So from a stem cell transplantation perspective, patient burden we see is extremely high. In the CAR T space, I'm calling it less invasive, but there's still chemotherapy, low doses. And I'll walk through a little bit of kind of what that process looks like. And so it's an area that I really wanted to focus on for my company is how can we actually decrease that patient burden. But overall, from a efficacy and safety perspective, CAR T may eventually offer a safer, more targeted alternative to chemoablation, and especially for patients that actually aren't candidates for the stem cell transplantation. So the challenge, though, is if we extrapolate our learnings from autologous CAR T cells in oncology, we have great clinical results. But we don't always see those great clinical results resulting in significant patient access improvements. So what I wanted to do now over the next few slides was share some of our learnings in oncology that I believe could be directly related to autoimmunity. And so what we're looking at on this slide specifically, this is the efficacy for diffuse large B cell patients in third line before CAR T cells and then after CAR T cells were commercialized. And so on the left side, we're looking at before CAR T cells. The treatment was salvage chemotherapy. And so the overall response was low. The complete response was less than 10%. And your median overall survival was roughly about a half a year. When CAR T cells were approved, we compiled the data, we look at this third line DLBCL patients, we're seeing complete responses greater than 50%, and we're seeing median overall survival beyond 2 years. So this magnitude of improvement in patient outcomes is typically unheard of. It transformed the way that we think about treating cancer. But the challenge is that the CAR T usage in oncology still remains frustratingly low. And so what we did is we looked on the left side at the numbers of patients that are treated based on doing some revenue-derived patient volumes. And then on the right side, we looked at how many patients could be treated based on the approved lines of therapy. And so on the left side, we're seeing roughly about 8,000 patients per year are getting treated with CD19 CAR T cells. But on the right side, that's suggesting that only about 10 to 20% of the patients that could get CAR T cells are actually getting CAR T cells. And so it begs the question, if there's so much patient need, how come there's this significant gap in patients that are getting treated versus those that could? And so we ask ourselves why. And at the highest level, if you look at the treatment journey for a patient, being treated with an autologous CAR T cell therapy is one of the most complex treatment journeys of any disease, any line, any geography. And the challenge at a high level of how this works is that essentially the patient gets their cells removed from their body. Those cells get shipped to a facility that genetically modifies those cells, grows those cells up, and then ships them back and are infused into the patient. That whole process takes over 100 days right now on average. And so during that time, these patients are sick. These patients are getting low-dose chemotherapy. It's a very complex treatment journey. And so what we wanted to look at was what are ways that the industry is looking at solving this? And so what I would look at, I think of it maybe 3 ways that biotech is tackling the problem. And on the left side, in the autologous CAR T space, there are companies that are looking at decreasing that time from manufacturing, from taking the cells out of the body to manufacturing the cells and putting them back in. So they're really trying to reduce the time for patients are waiting for their drug product. Another way that companies are looking at trying to solve the patient accessibility and patient experience is a platform called allogeneic CAR T cells. So this is, for instance, taking a healthy donor's cells, genetically modifying those and using those as the treatment for the patient. And then one that I've highlighted because it's near and dear to my heart is instead of taking the patient's cells out of the body and genetically modifying them, can we actually genetically modify the patient's cells in the body? So we're calling that in vivo CAR T cells. And that's an area that I can focus on a little bit to talk about kind of the up and coming future of how we're thinking about treating autoimmunity and oncology with CAR T cells. So from an in vivo perspective, we believe that this in vivo delivery solves a lot of the challenges that I've highlighted in the ex vivo CAR T cell therapy space. So what I wanted to highlight here was kind of the left side picture. That's actually a picture of a drug product that we have manufactured in our facility just outside of Boulder, Colorado. It has gone into a US IND, and we are now actively treating patients with this drug product. So this is a highly scalable, one manufacturing run can make hundreds, if not thousands of doses per patient's material. And then from a patient experience, there's no lymphodepletion. The vision of this product is that you walk into the doctor can give you this treatment off the shelf. It's similar to getting like a flu shot. You can walk home and go home, and the body then starts to do the manufacturing and cancer fighting drugs inside the body. Ultimately, what that results in is tumor and pathogenic autoimmune cell eradication through the generation of these CAR T cells. So we're eliminating the entire ex vivo taking cells out of the body manufacturing, and we're doing all of that within the patient's body. And when we started the company, there were two areas that I wanted to focus on to enhance the patient access with this in vivo space. The first was that we had to have a product that works. And so we spent the last 5 years really making sure that this product can actually go and target what we want it to target. So in our case, target the T cells, genetically modify those T cells to express the autoimmune fighting CD19 cancer immune fighting drugs. And we've done a very good job of that. But I did not want to just have a great drug product. We wanted to make sure that that product has the ability to be affordable and accessible globally for patients. So we spent as much time that we did on the science and making sure that we had a great drug product. We put an equally amount of time into making sure that it's manufacturable, that patients won't be waiting for drug product, and that the drug product will be waiting for patients. And so we have built out a facility that has a very clear line of sight for commercial and global accessibility for patients. And so what has that resulted in the in vivo space? I'm just highlighting Umoja's pipeline, but there are many companies out there now that have similar pipelines looking at both oncology and autoimmunity. And so we are now entering the clinic and entering that new phase of drug treatments in autoimmune diseases with in vivo CAR T cells. And so I'll leave you with the thought that we started off by asking what's tomorrow's opportunities in cell and gene therapy for scleroderma, and I would say that tomorrow's opportunities are here today.
Well, welcome to all of our participants, and thank you so much for tuning in to this incredible session. I want to first start off by thanking Dr. Elaine Karis and Dr. Ron Crisman for some fantastic presentations. And we have both of them here with us live, and we get to ask a couple of questions. And thank you all for your questions coming in over the line, and please feel free to continue to ask questions, and I'll try and pick them up as we go forward here. But a number of the questions are around whether or not this therapy might be a fit for an individual patient, and then what some of the other sort of challenges are to the therapy. So why don't we take a moment and just step right back in. I'm going to talk to Dr. Karis about what patients are included and excluded in a number of these clinical trials, and if you want to just sort of talk a little bit about how you think some of the different industry participants are thinking about the right patient for B cell depletion, in particular for CAR T B cell depletion.
Sure. Thanks, Luke. So for the clinical trial perspective, it's really important to, at this stage of development, because it's important to remember that these products are still in development. They're not approved products. And so at this stage, we want to enroll patients in the clinical trials who are able to provide us the information about how effective the drug or the cell therapy can be. And in order to do that, patients need to have a certain degree of scleroderma involvement in their body. So that can either be in the skin or in the lung, as the majority of the trials are looking for patients who either have extensive skin involvement and or lung involvement.
Great, thank you. Maybe just to sort of talk a little bit about it, because you touched on sort of some of the, what happens to patients as, upon therapy. Maybe we sort of step through. I mean, one of the, if you will, sort of, signature features of this, which is so exciting, is that it is a quote unquote kind of one and done therapy, if you will. Of course, there is some preconditioning prior to the administration of the cells, but let's just talk a little bit about sort of what happens immediately after. You mentioned two different side effects by their acronyms. Let's just take a little bit of time and sort of go through those: CRS on the one hand and ICANS on the other. So let's just talk through a bit more. Again, I'll have Dr. Karis talk a bit about what those two are and how they get managed, frankly, in the real world and also in the context of the trials.
Sure. So I think it's really important to understand that the B cell depletion that occurs with the CAR T and with the lymphodepletion is a very powerful B cell depletion. And so when the B cells are depleted so quickly and effectively from the body, this can result in these potential adverse events, the cytokine release syndrome and also the ICANS. And these are events that are, specific to CAR T. I mean, they can be seen potentially with other products as well, but they are adverse events that have been identified and are known to occur with CAR T. Now, everyone who comes into a clinical trial who receives CAR T is potentially at risk for these adverse events. The good thing is that these adverse events are not new to the cellular therapy world, because cellular therapy has been approved in oncology. And so we know a lot from oncology about how to identify these adverse events early and how to treat them. So when patients come into a clinical trial and they receive the CAR T, they are very closely monitored for usually the first 10 days or so in most trials. And during that 10-day period is when the cytokine release syndrome and the ICANS typically occur. And so because patients are being so closely monitored in the clinical trials, early symptoms can be quickly identified and addressed. I would also comment, for the cytokine release syndrome, we're typically seeing lower grade cytokine release syndrome, including what is called a grade 1 CRS. And a grade 1 CRS is a fever. It's basically a fever after receiving cellular therapy. And so I just wanted to put that in perspective for how these potential adverse events can show themselves and what can be done.
Thank you. Maybe just to shift gears, and again, we're talking a little bit more looking out into the future. But Dr. Crisman talked about gene therapy for T cells. So we're directly modifying the T cells in a patient's body to basically then arm that T cell to eliminate the B cells in that patient. Sounds sort of Star Wars-ish, but you noted that we're already in human clinical trials. If we just go back, what's the evidence from the animal model work which qualified Umoja in particular, maybe other companies for clinical work that this was safe and effective.
Yeah, I think if we look, it's really only been a year since Chet highlighted his clinical data showing that we can get very good B cell reset with CAR T cells, and that was really founded off of kind of early oncology data over the last decade, but then also the animal model data that you suggest showing that we can get very deep B cell reset in the tissues and the bone marrow with our non-human primate data, and that's across a lot of different platforms. That's with T cell engagers have now shifted into looking at opportunities within autoimmunity. We've got allogeneic, where you're either using healthy patient cells or you're kind of modifying cells to grow up a therapeutic dose. We've got those companies are in the clinic. They're going to be releasing data very soon. And then, folks like Umoja on the in vivo side, where we're directly genetically modifying in the body. Those clinical trials are starting now. They're very early, as Elaine mentioned. And so that, we ask, well, like, why are we going with the very sick, very, for a huge unmet medical need, and why aren't this preventative? We're working our way there. The oncology side is 10 years in. And I think we're moving a lot faster, but it takes some time to make sure this is safe, make sure that the mechanisms are working the way we think they are. And then we'll move our way towards earlier preventative medicine.
Yeah, thank you. So in the context of immune reset, what are some of the features that we're tracking to sort of convince ourselves that not only obviously we can follow symptomology, but when we sort of talk about this, if you will, something like nirvana of actually resetting the immune system and effectively, making an autoimmune disease patient now, quote unquote, have a normal immune system. What are some of the features that we're tracking to convince ourselves we're actually achieving that really dramatic, almost, if you will, kind of cure-like status?
Yeah, I can take a start, Elaine, and then you can jump in. But if you think about our immune systems, it's a really delicate balance. And when that gets shifted one way or the other, we see diseases start to occur. And so, for me, the thought being that when you see the loss of tolerance, and so really our body balancing our B cell and our T cell interaction, you lose that tolerance, and then you see the diseases start to show. And so the idea being that if we can figure out how to reset that, maybe we'll call it the bad interactions of the B cells and the T cells, we then will start to see the effect where the patient won't have the symptoms that they're showing. So we're really trying to get to targeting the root cause and not the symptom. But the symptom is what we're going to be tracking for a while to make sure that our hypothesis of really doing a tolerance reset and kind of going back to a time when our B cells and our T cell interactions were healthy is what we're targeting. We'll be looking at that though from a symptomatic perspective for the short term.
Great. Well, great. Unfortunately, we have exhausted our time today. There's so much more to talk about in this incredibly exciting area, but I want to sort of finish by once again thanking Dr. Karis and Dr. Crisman. Fantastic presentations. Thank you so much for sticking around for the question and answer, and I'm sure there'll be more follow-up from our patients, and we'll look forward to being able to answer that more one-on-one. But thank you again for your contributions today, and goodbye for now.
Thank you, Luke. Bye-bye.
Thank you. Bye.